Hello, good afternoon to everyone and welcome back to the channel The Upcoming Doctors. So in this session we are going to discuss about the physiology of blood. So let us get started. Here we can see that is hematology. Hematology normally means the study of physiology of the blood and the blood disorders. And normally we do the blood test. So the blood examination will reveals two fundamental aspects. Normally the bone marrow will be present and the bone marrow will produces all the blood and the blood cells. So, if the bone marrow is producing the number of the cells in adequate number or not, and produced cells are developing in a right manner or not, these two fundamental aspects will be revealed under the hematology. So, if the bone marrow producing adequate number of the mature cells of the hematopoietic lineages or not, this is the one point. Another point is if the development of each hematopoietic lineage is normal or not. So, coming to the blood, it has normally cellular component and fluid component. It has two components, okay, right? It has two components, and the cellular component will consist of different formed elements. What do you mean by the formed elements? This are RBCs, or otherwise called erythrocytes, WBCs otherwise called leukocytes, platelets otherwise called thrombocytes. These three comes under the formed elements. And coming to the fluid component, it will be like plasma. Okay, the plasma again consists of the water and the solid particles. 8% of the solid particles will be present, 92% of the water will be present out of the 100% of the plasma, and the 100% of plasma itself contributes about the 55% of the total blood volume, and remaining 45% will be constituted by the cellular components, nothing but the RBCs, WBCs and platelets. So the plasma proteins will occupy measure of the solid particles in the plasma. And coming to the blood definition, it is a liquid connective tissue that fills the heart and blood vessels. Obviously the blood can be seen in the heart and blood vessels, no? And the normal blood volume, this is very important, right? The normal blood volume in our body will be 5 to 6 liters, and it constitutes about the 8% of our body weight. Okay, if suppose your body weight is 50 kgs, like 8% of the 50 kgs will be your blood volume. Okay, normally the blood volume will be 5 to 6 liters in our body. This is very important, remember the blood volume. And when blood is collected in an anticoagulant tube, because you took the blood and we need to add the anticoagulant, right? Anticoagulant like heparin we can add, and then allow to centrifuge. Centrifugation is done, and then we can observe the three layers are distinctly visible. This here we can see the three layers, right? This is the plasma, which is uppermost layer, and it constitutes about the 55% of the total blood volume. And then we can see the buffy coat, which is less than 1%, and the red cells around 44 to 45%, which can be the bottom layer, that is the third layer. This is the first layer, and this is the second layer, buffy coat, and third layer is the red cells. Okay, and coming to the from up to down, that's I written here. That's a plasma layer, 55% we can see here, and then buffy coat, that is less than 1%. It consists of WBCs and platelets. So WBCs and platelets are present in this buffy coat, and this buffy coat is clinically very important, because normally in the buffy coat, in normal conditions, normal individual, it consists of the WBCs and the platelets. But in abnormal conditions, if something becomes abnormal, then definitely the abnormal cells are present in the buffy coat. So by forming the centrifugation, we can observe any abnormality is present, otherwise everything is normal or not, we can find out like that. So buffy coat, the thin middle layer, this is the buffy coat, the thin middle layer that separates the upper plasma layer is present, lower the red cells are present. After the centrifugation, we can observe this three layers. Okay, and this is again very important, and clinically it is important. I already said in abnormal conditions, the abnormal cells are found in the buffy coat. Okay, and we can see here, one minute, we can see here, yes, this is the component of the blood. So we can see the plasma here, and less than 1%, this is actually less than 1%, that about the platelets and white blood cells can be seen, and 55% of the plasma, which is uppermost layer, and the red cells, which is around 44 to 45% can be seen. So these are the components of the blood. Okay, and then again coming to the third one, this is the last part of our session. So the plasma proteins, already we saw the plasma consists of 92% of the water and 8% of the solid particles, but again in that 8% of the solid particles almost will be constituted by this plasma proteins. Okay, the solid particles, mainly three plasma proteins are present. These are albumin, globulin and fibrinogen. Okay, this can be separated by using the salt separation or paper electrophoresis or the ultracentrifugation, and these are the methods to separate the plasma proteins. Okay, to separate the albumin, globulin and fibrinogen. Again, you should remember one thing, the A by G ratio is very important. Clinically it is important, actually it should be normal, otherwise it can indicate some diseases. Okay, and this is very important. Now you can see here, this is the plasma is equals to serum plus clotting factors. Normally plasma will be consist of the serum, which is the liquid component, and clotting factors. Okay, clotting factors also will be present in the plasma along with the plasma proteins. So now what is meant by serum? Serum will be plasma without the, this will go there, and minus C. So plasma without the clotting factors is called our serum. And coming to the functions of the plasma proteins, briefly, the albumin is the main plasma protein that maintains the oncotic pressure. And what do you mean by the oncotic pressure, right? What do you mean by the oncotic pressure? The osmotic pressure which is maintained by the plasma proteins is nothing but the oncotic pressure. And coming to the globulin, it maintains the immunity. Okay, and fibrinogen, this is an important plasma protein which can do the blood clotting. So the fibrinogen is converted into the fibrin. This is the ultimate step of the blood coagulation. So the fibrinogen is very important. And coming to our last topic, this is bone marrow. Okay, normally this is the site of normal hemopoiesis, or otherwise called hematopoiesis. Okay, hemopoiesis otherwise called as hematopoiesis after birth. And it consists of, bone marrow will consist of three types of marrows: red marrow, yellow and the white marrow. Okay, mainly these two can be seen, red marrow and yellow marrow. So let us see in detail the red marrow. Red marrow is the active site. Okay, this is important, active site of hemopoiesis. Hemopoiesis means, poiesis means production, and hem means blood. Okay, the hemo means blood and blood components. So production of the blood and blood components is called the hemopoiesis. This will be occur in the red marrow, which is very active site, and present in, this will be present in the almost all bones of the body at the birth. Okay, and as the age advances, that it is replaced by the fatty tissue and slowly becomes inactive, and it will be active only to the places at the age of 20 to 30 years, you will get. Now then the red marrow will be active only in the axial skeleton. What do you mean by the axial skeleton? Now the skeleton will be like this present, no? So whatever will be present in the middle, okay, in the medial side, this is called axial skeleton. Almost I'll say this is the skull will be present, no, and here the sternum, ribs will be present, and also the girdles, two girdles, we have two girdles, like girdle, it literally it mean the crown. Okay, crown. Okay, crown. So we have two crowns, which means two girdles. One is the pectoral, this is not, you remember we have two girdles. Okay, two girdles will be having this called, one is the pectoral girdle, another one is the pelvic girdle. So we have two girdles. So this all constitutes the axial skeleton: skull bones, vertebra, ribs, sternum, pelvic and the pectoral girdle. Okay, you remember this. The red marrow will be very important, and red marrow is the active site. Okay, this is the important point. The red marrow is the active site of the hemopoiesis, and it present in the almost all the bones of body at the birth. As the age advances, the red marrow is replaced by the fatty tissue, and 20-30 years you will get means the red marrow only limited to the axial skeleton, and it will be active only in the axial skeleton. And coming to the yellow marrow, it doesn't take part in the hemopoiesis, and the fat cells will be replaced by the replaces the hematopoietic cells in the bones of hand, feet, legs and arms at the age of 20 years. Okay, this is the 20 years. You can be see that the yellow marrow is also, it is replacing the hematopoietic cells into the fat cells. So at birth only we have more red marrow, and some of the hematopoietic cells will be replaced by the yellow marrow at the age of 20 years. This is all about the introduction of the blood session. So please share, subscribe, like this channel, and whatever your doubts are present, please comment me in the comment section so that I can answer to them. And follow the Insta ID, that upcoming doctors, and join the Telegram also, the upcoming doctors, for the notes. Thank you so much for your support. Thank you.